density water.
Yeah, there's a little bit of shimmering.
There we go. Sorry, I can't hold that steady.
No problem.
Dan, if you could tilt down a little bit for me, please. Seem to have my hands busy.
Yeah, we are seeing some shimmering in the water.
See the center there with the shrimp.
Cute.
Great face.
And what is that inside? Something's moving inside.
So this is purple, and presumably a purple octocoral, but it looks like it's almost encrusting, and I'm wondering whether it's more like a stoloniferan rather than Victigorgia, which is an actual upright gorgonian.
Coming for a slightly wider shot.
Bridge ROV nav.
All right. Video's clear.
Okay, just above it and to the left, there was a soft-bodied thing, looked like
Just above and to the left?
Yeah,
Okay, we'll
like
take a
right
look
behind,
at that.
Right behind the fish. Just caught it as we zoomed out.
We'll find out what it is.
Yeah, tilt up a little bit there.
Now, this is Bruce again from NOAA Fisheries. If we have a moment, there are a couple of other odd things about these green-eye fish.
Again, Levi.
One of them is that they're synchronous hermaphrodites.
Oh, yeah. Might want to explain what that is to the general public.
Yeah, so they have both testes and ovaries at the same time. That doesn't mean necessarily that a single individual will reproduce, that these sort of self-produced, self-fertilizing eggs. But we're seeing so many of these together, so many of these green eyes, that seems highly unlikely. But they have the capability of acting either as males or females at the same point in their life, which is a very unusual reproductive strategy for fishing. The other thing is that at least one of the species, and probably most of them, has a light organ on the ventral part of the body that light's produced by symbiotic bacteria, which is a way that a lot of fishes produce light. And since the light organ is on the ventral surface of their body, they're not using it for, probably not using it for camouflage or for attraction of prey, but it might be something that's used in this low light or lightless depth for species recognition. That's speculation for the function, but they are among the animals that produce light.
Yeah, I'm trying to see what kind of distance.
And Bruce, in terms of the synchronous hermaphroditism, they have sperm and eggs at any one time. Don't fertilize themselves, but could fertilize their sperm with another green eye with eggs and vice versa. Is that what you're saying?
I'll call it in.
Okay.
Bridge RV nav.
That's correct. That's correct. They don't really know much about the actual reproductive behavior of these things because they live so deep that people haven't kept them, to observe the spawning. So there's a little speculation there. A lot of fish are hermaphrodites, but they're what are called sequential hermaphrodites, and good examples are the wrasses and parrotfish on coral reef, where they'll start off light as females, and as they grow, then the larger ones will change to males. Or the anemone fish, the Nemo fish, do the opposite. They'll start off as males, and then as they get larger, the largest ones will turn to females. But the green eyes are unusual because they're both at the same time.
Thank you, Bruce.
Watch lead this pilot.
Yes?
We've just noticed in the last couple minutes here that the current is now coming from the east. So in the span of maybe 15 minutes, the current went from a westerly to easterly, coming from, that is.
Okay, roger that. So we've had a shift in the current, and just over the last 15 minutes, right, from west to east? Is that what you're saying?
Yeah, it was coming out of the west.
Was coming out of the west.
I should be saying it was setting to the east, and now it is setting to the west.
Okay.
Does it feel pretty strong, or just a light current?
Yeah, I've got a steady. Well, let's see, what have I got on here?
Yeah, but there's some
Steady,
sea
like 30%
stars,
now.
sponges,
Lots, yep.
green eyes.
Use it.
Okay. Getting closer to you now.
Okay. Now I'm going to start pushing ahead.
Yep. Yeah, so I think during that ship move, Sirius must have started to get swung around the other side of the ridge. Which
Okay.
didn't help. And then also the ship started moving at like 310 for a bit.
Okay.
Oh, wow. Look over there. In front of the ROV? I'm serious?
Yeah,
Tons of these sponges.
yeah. Yeah, if you look
So,
down, yep.
This is Chris at UHBCC. Obviously this demosponge, this ribbon-like demosponge, is the dominant animal at the site, and I don't think we have a biological sample yet, if I'm not mistaken.
Okay.
I think it could be Posalastra, but I'm not really positive, and so I'm wondering whether or not this would be a collection candidate to get a piece of one of these white sponges.
Yep, I was going to ask you that, Chris, if you knew it well enough to get it down to species. But since it is really one of the more dominant organisms in this area, I think it would be worthwhile to get a sample of that. Let me ask the pilot about that. Okay, pilot, we would like to get a sample of one of these white sponges, please.
Okay.
But can we also look at that crab in the distance as well, please?
All right, Levi, I think we can bring the ship to an easy stop here.
Yep, we'll do that, Diva.
Okay, I'll stop the ship.
So again, also in the let me tell
Bridge, are we
pilot.
now?
Tara, let's take a quick look at the
I'm looking at the wrong one here.
Slow down, then I'll
We
slow
can
down,
go ahead and
try
bring
and
the
get-
ship to
Okay,
an easy
roger
stop
that.
here.
So go ahead and zoom on that.
Oh, wow.
Crab moving along here.
Spiny crab moving along. Look at all those spikes on him.
I'm just readjusting my
So
head.
this is definitely a stone crab in the family Lithodidae, and given the length of the spines on there, I suspect it's probably something in the genus Neolithodes.
Thank you.
So, Chris, did you say stone crab?
Yeah.
Like the stone crabs we eat in Florida? Don's
That's
shaking
correct.
his head. I know. I'm just asking.
And the actual name, the family Lithodidae, just like Lithistid,
Yeah.
the name is derived from rock or stone.
You both lined up. with
Very
the bridge
cool.
now.
Okay. I'm just going to push out a little bit so I'm not.
Diva,
right
do you
on the
remember
edge.
seeing this on the
Now I'm trying to think.
on
I
Leg
believe
1?
What I'd like to do to get a sample would be maybe go over to the
We
left
did
a
see one of these on leg 1, and Mary Wixton did identify it as such, as what Chris said.
Yeah. So I'll just push ahead here a little bit.
Well, what
If
the
I
pilot's
try to do it up here and I take a
pilot's
sample of the
doing
sponge,
is he's maneuvering the ROV over to the west side of this ridge, and going to be pointing eastward into the current. So it's easier,
to collect.
to do that if you
easier to
maintain
Position.
stability
Yeah,
of the
position. So the,
ROV.
Lights are all in, right?
Folks, if you take a look at the Sirius image, you will see why it is that this sponge is the dominant organism on this dive. That is a fabulous image from Sirius. I happen to personally really enjoy looking at the ROV from the perspective of what's basically a clump weight vehicle above the ROV, the Jason 2 and Medea vehicles that Woods Hole Oceanographic Institution
So I'm not gonna, if I'm
operates
pushing against
also,
the skids, I'm not going to be able to extend
operates
the
is that
correct?
they have that
But,
same capability, and you can get the context of the geology, or in this case the biology, And look at all the shadows from those sponges.
And Pilot, don't worry too much about trying to get an entire specimen. If we can get a piece of it, that's fine. It'll be enough for us to be able to identify down to genus and species. So don't be too concerned about trying to rip off an entire specimen.
Understood. Thank you.
And here comes our crab. That's a cool-looking crab.
It is very cool looking.
And several of us were thinking if you could get a piece of the sponge with the green filamentous things attached, that might help about that.
Okay. All right. Okay, Pilot, did you catch that?
No,
so we're gonna increase the level of difficulty on this collection for you, and see if you can get a piece of the sponge that has some of that green filamentous stuff. But I don't see any large pieces of sponge with a green filamentous. But we can take a look at it for a couple minutes and see.
We're seeing sea urchins as well now.
There's one right in front of me. It's not
and
very big,
white sea star and some little red squat lobsters on the seafloor.
Yeah, Jeff, you're getting in on it. I see what you're going for.
I think it's out of reach. Yeah, it's out of reach.
For those of you who study these organisms that are not attached to the bottom, I know you're secretly wishing, oh, if only we could get the green eye. Oh, if only we could get the lobster or the crab or the shrimp. It's virtually impossible.
True enough. Maybe while we're getting in position here, a couple of minutes, if we could ask Chris Kelly from University of Hawaii Oceanography Department, and he is the person in charge of the Expedition Control Center in Hawaii, if he might just give us a real brief overview of the objectives of this dive. He was the proponent who submitted the dive plan.
Okay.
So right now your port toe is touching much before your starboard toe does.
Yep.
I wonder if Chris has gone out for lunch.
Too early.
Too early.
Unless,
But he is allowed to take breaks.
That's true.
The green.
Maybe we'll hear from him later.
So this is an area that was proposed to dive on because of the potential for having precious corals. And there had been anecdotal reports that precious corals existed here. And since precious corals are part of the managed fisheries from Guam and the Commonwealth of the Northern Mariana Islands, we wanted to take a look at this site and see if, in fact, there were precious corals here. And so far, we've seen one species of precious coral. So the bamboo coral that we saw earlier is is an example of a precious coral. But in general, it's not. There's not a lot of precious coral species on this at this site. So that you're looking at right. Let me get to the Pilot here. So Pilot, what you're looking at right now is another species of sponge. It's that lithistid, but yeah.
But what we want are the ribbon sponges, right? Or the
what
white
we want is the ribbon sponges. Chris, have we gotten any lithistids?
Right now, so it looks like maybe you're
Oh, he's not here. David, do you know if we got any lithistids on leg one?
So this is the wrong kind of sponge with green stuff on it. Okay. Let's go get the right kind of
Well,
sponge.
here's the thing. If you're a sponge biologist, it's not the wrong kind, because that's another one that's very interesting. But yeah, let's focus on getting the thin fan sponge, please.
Okay, you got it. I think I see another candidate just up here forward a little bit.
Okay.
It's got green stuff on it.
The one up just at
Yeah,
the top.
you do.
Nice.
Yeah, there's
Perfect.
Yeah. There was another one at the bottom too. So if this one doesn't pan out, you can try the bottom.
So as we are waiting to attempt to do this first of our 2 permitted biological samples, and I'm not sure whether this constitutes both.
No, that's a commensal. That'll be a commensal on it if it's attached. I'm already thinking my strategy here.
What I was going to say is this is the location where we're diving is on Ferdinand de Medinilla. It's a very small island, second smallest island in the group. Basically, only there's a bank that's a little bit smaller than this that rises above sea level.
And if I look at the
and
port side rock box there, I don't think you're going to
the
be able to
objective
extend
of
the door right
this dive is to look
The
at
other thing is that with the current,
precious corals
if we just
in the
were
depth
stationary,
range
you
that
might-
we
you might
are
just float away.
diving
Let go of
in,
it. So
which
we
is
could-
from
we could
about
give us some upward thrust.
527
Well,
or so
box.
meters up to
As you come up, setting out a little bit of
97,
momentum upward,
something like
slow
that.
down, you let go.
527 to 497. And that is the range in which precious corals, which
I
are
think it sounds like a-
commercially valuable,
The drawer
have been
open.
identified.
The drawer can
So
open a
far,
little
we
bit, I
have
think.
seen 2.
But yeah, probably not
We've
all
seen,
the way.
the bamboo coral. So,
Okay.
yeah, I don't think
Okay.
we
So you
And
just
we
grab
did
it,
see a black
you grab
coral.
it, you have it in
Oh,
your
that's right. We
claw,
did see a black coral.
At
You're
the-
right.
kind
Right,
of-
Patty. You did.
Kind of right at the beginning of the dive.
And if we are concerned
most
about
of
it
the
getting blown away in the current,
corals
like,
and
put it in auto depth,
sponges, such as
bump
the
it up
ones
a
we're
bit.
looking at now,
just
crinoids,
meter or two,
which are
you'll get a little bit of
a
downward
stalked
flow
animal
into the
that
box.
has sort of feathery
That sounds
arms
good.
sticking up
We're
from the
nice and
top of
stable,
its
so
stalk.
this is a good
All of
place
these
to
animals
do
are
this.
filtering
So if we bring hydraulics
plankton,
You got it.
small animals out of the seawater.
So, pilot, this might be a brittle sponge when you go and catch it, collect it.
You're
So
just
we
making
chose
everything easy for me.
this dive in collaboration with the person who initially proposed it. So one person proposes the dives, and then we have
Nice eyes
teleconference
on-
or video conference with a large group of shore-based scientists who all comment on these dives and put in their 2 cents. Also add objectives that they think might
if you could
be
just keep an eye
appropriate
on
for the
serious
sorts of
camera, or
dive
settle
that we
out, but-
choose each day. And we all, as a
dive.
group, make up a final dive
Yeah, you have
track,
hydraulics?
so the
You have
the line
hydraulics.
across the seafloor that we're going to follow. So we're basically
Are we
diving
full light
on
on
the
Minizus?
very tip top of a narrow ridge on the flank, the southern flank of Ferdinand de Medinilla, and hoping to be able to find enough precious corals and things to give sort of an inventory of the species that are existing there. And right now what we're doing is we are collecting what the NOAA Ocean Exploration
What grip force are you in
group
right now?
would call a dominant species in this region. So as you can see, the seafloor is just crusted everywhere possible almost with these white ribbon-type sponges. And so one of these will be our sample, our first biological sample on this dive. So for scale, the fingers from the metal part of the manipulator arm that you see is about
I'm gonna
9
give you a
inches
little
or
bit of
so
zoom on Minizus.
long. And when it's fully extended like that, it reaches out
Hold it
to
right
maybe 10 inches or so. The pilot has pressure control sensitivity, so as he is taking a sample, this sensitivity is good enough that he could pick up an egg without cracking the shell. But also, if he's trying to get a good grip on a rock,
I couldn't close the
he
jaw
can
on
really
deck until grip force
pressure
the other
down.
day.
Basically, he can
Losing
feel
pressure,
with his
pressure at
hand
right now?
the sensation he would have if he were actually
1500
picking that rock
RPM,
up
so
Okay.
off the ground
Good.
surface.
That's grip force 5.
So these sponges may be kind of crunchy. They're kind of brittle. So
Force 4.
we'll see what we can do with this.
So do you think fragments of it might
Yes.
break off?
Yeah,
and,
that's
and
what I'm
maybe
thinking.
Could pick those
That's
up?
what I'm thinking. They may break off. And again, for an identification, we just need a representative sample of it, because what we'll do is we'll take a piece of it and make a section of it and look at it under the microscope and see what kind of architecture it has. But we'll also take a piece and dissolve it in bleach, just common household bleach. And what will be remaining will be these microscopic skeletal elements called spicules, and the spicules are often very diagnostic
Right now your
for
top fingers
to
are
help
gonna
us to
hit
get down to to species of sponge as well.
Can you,
So these the sponge, which Chris and I both think might be a
it
pacastrelid,
up and
will have
feather it
very
in.
distinct
maybe a
spicule
little more
types as
precision
well.
with the grab.
Is the name of a sponge indicative of anything about the sponge, or is it the
Well, that's interesting.
person who
Okay.
found it first?
For those of you who took Latin way back when, what does PAC stand for? Think of pachyderm. What's a pachyderm?
Oh, hey, we got a piece. We got a piece
no.
with the commensal. Let's go for it.
No.
No, I'll tell you what, this is really tough to do.
Yeah,
It's a
it really
brittle
is.
sponge. We got ripping current,
Yeah.
and it's really tough to do. So pacastrelid, Strella's star, pac is kind of thick skin. So what these sponges have are basically a skin or an outer layer that's loaded
really
with
tough grab.
spicules, these microscopic siliceous skeletal elements that look like stars.
Aha.
Little starburst. So we have on the chip a microscope, and I've got some slides and cover slips, and I'll go borrow some bleach from the stewards in the galley and try and identify it. So what happens with these samples after they're collected is we put them into an appropriate preservative, mostly alcohol, ethanol, and we take photographs, we make notes about the organism itself, and then these are all labeled, of course,
thin,
and then the
fragile film
biological
without
samples
much
go
depth,
to
you
the
know,
Smithsonian.
height.
And so you'll be able to check and see the mechanism that you would use, the procedure that you would use to bar these. But these samples that we collect are for the scientific community to study. So it's not they're not my sponges. They're not Chris's sponges.
They're certainly not my
They're
sponges.
certainly not Patty's sponges. But there are groups, I have colleagues who work on this group of sponges, and so they would be able to put in a request to the Smithsonian to borrow the sponge. And the same thing happens with the rocks. The rock samples are going to Oregon State University, correct, Patty?
That is correct.
And
we
that- yeah.
just kind of knocked this loose a little bit. And, you know, if folks are interested, as Shirley just said, you know, we've got a microscope here on the ship. All of the pictures that we take of the samples that we get will be available for people to look at on the NOAA's
Ocean Explorer website. Yep.
Yeah, so if you hear
right
something like what Shirley said about, you know, being able to see the star, I bet you guys are all sitting out there going, oh, I wish I could see that. Well, you can. So that's all available on
Ocean Explorer.
oceanexplorer.noaa.gov. We might not have it up today.
No, it won't be up today.
But all that kind of stuff will be available for everybody to look at.
So in addition to the biological samples that are going to to the Smithsonian, and the rocks samples going to an archival, like a museum at Oregon State. There's a third sample that we're taking, and this is of the biological sample. So a very small piece of each biological sample is being put into a special preservative to look at molecular work, so DNA, for example. So those are going to an organization called Ocean Genome Legacy at Northeastern University. And so Smithsonian, Ocean Genome Legacy, and Oregon State University are partners of NOAA's Office of Ocean Exploration and Research. And this is a way for us to be able to have these samples that are properly archived. We know where they are at any point in time, and everybody can, anybody who's interested in studying them can put in a request to study them.
We can keep an eye out for a bigger fan somewhere.
Okay, pilot's loosening up. He is not going to let this get the better of him. They're tough.
I am going to let this particular sponge get the better of me.
Yes, there's plenty more out there.
Situationally, this just...
Yeah.
I had 2 pieces come off, and the current just immediately takes it away.
Yeah, understood. This is really tough because I said these are like, you look at they're really thin sponges, brittle. So as soon as they're dislodged from the substrate, they're free swimming, and in this current, they're really moving along. I mean, you can see in the upper part of the screen, you can see one of those filamentous organisms that's just really flapping about. So it's very challenging. So we'll let the pilot decide where he wants to be with the vehicle to get him in a better position.
Probably someplace that is a little flatter than right here on the slope.
Yeah,
would, if the pieces break off and they get blown around, maybe they wouldn't get blown quite so far on the flatter bit.
Yeah,
it is
hard to tell.
And our pilots out here come out on all these, each of the cruises has some trainee pilots, but also some people who are super experienced, have been out here on tons of these cruises, and so they are. I've been incredibly impressed sitting on shore watching them pick samples up. They do it so fast.
We haven't recorded that as a sample yet. Yeah, we do it once it's in the box.
See what Jeff says. Jeff, why don't you get lined up at the ridge?
No.
Yeah. Wow, the current is really...
Is it picking up?
Cooking now, yeah.
Yeah.
I just... Hang on.
I mean, I'm above the... You were behind the ridge. I'm above it, and I've been getting blown around quite a bit.
So in the meantime, as we're doing this, we're also getting observational information from our scientists, our land-based scientists, who are seeing urchins and bamboo coral, other octocorals. ASR.
Looks like I'm going to have to sidle along it here. So it appears to be about 30 degrees or 45 degrees to port of me right now.
Okay, so almost
350, something like that.
Okay. It's almost just about north, huh?
Yeah.
Thinking a 30-meter move or 20?
Let's do 20.
Yeah.
Just see if we can stay on the ridge and then...
You guys ready for me to call it in?
I'm ready.
Okay.
Okay, we're seeing more bamboo
Bridge,
coral.
ROV nav.
Some green eyes up the top of the screen, and one of those little
bearing 350
mini caves
degrees,
formed by the rocks.
0.2 knots.
So 20 meters, 350 degrees, 0.2.
Good copy, Bridge.
Okay.
So what we're trying to do now is survey this area and see if there's a place where the ROV can position itself to make it a little bit easier to collect this very brittle but most abundant sponge in this area. Nav, once you get a chance, can you let me know how far we are away from waypoints 2 and 3, please?
So you're just staying put right now with like
Watch lead, this
50
is
to
nav.
60% forward, huh?
It's crazy.
You are 230 meters away from waypoint 3. We just passed waypoint 2, and we have like now almost hour and 48 minutes left of the dive, looks like.
Okay, thank you very much.
What speed do we need to maintain right now to complete the dive?
Pilot, you're looking at, we got plenty of time. Actually, is that correct? A little over 0.16 knots.
Oh, 0.16.
Yeah.
All right. So, I mean, if we continue at 0.2.
Should get close.
Yeah.
Yep.
All right.
Let's see here.
Nice sea star.
No,
Can
hold
we
on.
zoom back in high pack when you're ready?
Yeah, I had the wrong numbers, pilot.
Okay.
We're only 227 meters.
Oh, okay.
So yeah, you have
Okay.
0.07,
Okay.
half of that. So I was, yep.
Cool.
So we'll make it.
Yeah.
I was wondering why it was so different.
Yeah, and we have time to stop and smell the
Yeah.
sponges.
Sorry about that. Yeah, we have an hour and 42 minutes of bottom time left and 220 meters to get to the last waypoint. So
Thank you.
Did we get one?
No, we're still working on that.
Yeah, my
They're
hand's
very
going around
brittle.
too.
So I wonder if what we've experienced is a change in the currents due to tidal change, because we had basically 180-degree change in the direction of the current, and now it's probably as strong as it was first thing this morning.
Yeah.
And we're 4.5 hours later. Go down. Here's another. And try and get this big fella right here.
Mm-hmm.
I can land kind of flat.
Just the one just to your left, is it what you're talking?
Bottom
Yeah.
left.
Yeah, that would be good.
Wow, that looks good.
You want to try to sample the sponge at the bottom left?
That's what I'm thinking.
Okay.
Yeah.
Hey, Levi, let's bring the ship to an easy stop.
Okay, will do. Bridge, ROV nav.
Actually, that move might be finished
ROV
already.
nav, this bridge.
I was just going to ask you to stop the ship, but I think it already is.
Yep, we're already stopped.
Good, perfect
Thank you,
timing.
Bridge.
That was some expert navving, Levi, right on top of the sample.
The current really seems to be moving along the little
Oh,
green-eyed
gosh.
fish, too, as they are.
Yeah, it's just ripping. That's a ripping current.
Dan, will you tilt the camera down a bit for me?
Got it.
I need to be able to see my skids.
How's that?
That's great.
I'll keep your camera for now.
Okay. Maybe better.
Pushing the vehicle off your toe.
Yeah. I don't. So now the issue is I have nothing to push into, and so it's blowing me backwards.
Oh, wow.
If I push forward, I skid forward.
Give yourself a little
All right,
more
so the current's really pushing.
downward.
Oh, I'm already
Pushing
The D2 back. Okay, so he's trying to kind of get positioned.
So what the pilot's trying to do is get himself stable on the seafloor to get close to that
I'd actually recommend
piece of coral
maybe
to
backing
sample
up a
it.
bit
And
too.
there's no sediment. You know, there's nothing for him to push against, and he's got a big current right in his face.
Is the camera tilted the whole way down?
No, I can give you more.
Okay, but there I am right at the front of the skids. So
Yeah, I think that's great.
that's a good spot.
Yeah.
If it'll settle in there.
You can, on either side of the screen now, you can see the sample platform extended a bit. The cameras are the camera is pointed way down,
Can you come off
almost
the
vertical,
boats at all
so he
without
can
moving?
see the skids.
No. Well,
Of the
it would
ROV.
Just, it would, no.
Okay, so we're about
Oh,
80%.
wow. Okay.
But I'll keep an eye on it in case
Oh,
we
wow.
fall.
Okay. Our pilot is
Well,
ready.
we'll All right.
Gonna try for it. Here he
Hydraulics.
goes.
Ready for hydraulics.
Okay, we've got hydraulics.
Okay.
And
Hydraulics on.
I'll give you the mini Zeus view.
So one of the dilemmas with this type of exploration that's telepresence based, and really that contributed to the decision that had been made prior to about a year ago to not collect any samples, is whether or not the goal is to just kind of go along the bottom, take video imagery, and
just
be
document.
able to, and document. And then it became clear that for some of these areas where we were exploring, we couldn't get a good feel for what the organisms were unless we had a piece of it. And so even though the Deep Discoverer has been equipped with the ability to collect samples, it's only been for the last year or so that samples are being collected. And so part of the consideration is that, how far along do you want to get during a dive? You're going to get like 800 meters or so, and we all acknowledge that no matter what system you use, to get yourself in position and do the collection can take a fair amount of time, because you have to position the vehicle, you have to take into account that there are currents, that the organism you're trying to collect is either squishy or brittle. Okay, you're good. You're good.
Got
Very good. Very good. Very good. Very good.
Out. So
Oh my gosh.
we'll get a close-up
Yeah.
view in a minute so that we can see what that looks like and
Yeah,
see
we're
if
good.
we got any
Oh,
of the
we're good.
green.
I can see it. We got green.
Got it.
Good.
Yep, you're
Great.
good.
Okay, hold that. Thank you.
You don't need to mess around with it too much while it's still floating free.
So the for folks at home, the fingers of this manipulator are about maybe a little bit more than 3 inches long. So imagine your fingers. If you took your index and your middle finger, you know, that's what you're trying to hold on to this thing with. As I said earlier, if anybody's just tuning in while we're sampling, our pilot has tactile feedback from this mechanical arm, and he is able to pick up pieces as delicate as an egg.
As this. as this. This is very delicate. Okay.
Now, the biological samples go in these white boxes that you see, and what happens is that the sampling sled on which these boxes are attached slides out from beneath the ROV, and a sample will be placed in these boxes and then slid back so that the chamber is closed.
Wonderful. Good job, pilot. Really good job, all of you.
Thank you, pilot.
Got it.
Excellent.
Nice work, Jeff.
Excellent.
Thanks, everybody.
Thank you. So that was going to the port out board. Now port.
Give you mini Zeus to stow.
Oh, it's specimen 2, even though it's bio.
It did not.
1, 2, 3, 4. Okay, sorry. Okay. Thank you.
And then actually, if you can rotate your jaw 180 degrees, try to stow it with the coral cutters on the inside. Perfect.
So we just
there we go. Perfect.
kind of keep the video image on the general area where the sample was collected, so that the scientists here on board can describe it sufficiently so that anybody who's interested in the sample has metadata or sort of general information about the substrate, what it looked like.
How's that?
You know, what time we collected it.
Okay, I don't understand what's happening.
And we get a good view of the area where the sample was removed from.
Clear. Okay.
To put it in context.
I think what it was, I'm holding the index button too long.
Yeah, if you do that, then it.
Yeah, it's a long.
The mini master will actually take the form of the arm.
Okay.
Fight against you.
Yep, that's exactly what's happening. I was holding the index too long. Yep. Okay. All right.
Okay. Looks like our pilot has taken a big sigh of relief.
Yeah.
Whoa, that was a tough collection.
It's a pain collecting these samples, isn't it?
Alrighty then.
Okay. So now shall we go cruising? Let's go cruising.
Yeah, he's getting ready to go.
Which way? I need to turn left and lift up. Okay, sir.
So you folks, if you look at the Sirius view, you can see the cable is behind it, but
left.
you also notice that Sirius is pretty much right on top of it,
Ahead.
which means that we can. It's easier to maneuver the ROV and make tracks.
Fighting current. Fighting current.
Matt, can we move this up?
Wow, yeah.
Can we move this
80 to
up
90%.
so we can see the track
and I'm getting
better?
pushed down as well.
I just want to be able to see the ship and kind of.
down.
Oh, okay. Is that nav?
Okay. I'm trying to push ahead. There we go.
Navigator. This is Science Lead. I'm wondering if we can see, if you could move the ship's position, center it a bit more on the screen, or maybe even kind of up toward the top, so that we can get a view of how we're moving along, the relative positions of the ROV and Sirius and the ship. Thank you.
Yeah, I can't quite turn that way, or I just blow away. So.
Well, Nav, how do you feel about it? 20-meter move.
Yeah, if the pilot's ready, it seems to be quite a bit of current.
Yeah.
If he can make way, then yeah, I'm happy with doing that. 20 meters. What bearing you guys talk about? Due north?
And
due north.
look at all these sponges.
I know. Look at them all.
I don't know.
I'm trying to like be, reserved and not be like gushing over this, but there's like.
I'll call it in.
Oh gosh.
So we.
Yeah,
no problem.
So we
Bridge,
study
ROV
these
now.
areas called sponge grounds, where you see pretty much monospecific, the same species of sponge in large
Ready for another
numbers.
sit move. 20 meters.
So this
bearing
would
due
definitely
north,
count as a
0.2
sponge ground.
knots.
20 meters, due north, 0.2.
And in much the same
Good
way
copy.
that corals, soft corals, gorgonians, stony corals provide habitat for other organisms, we've already seen that these sponges are also providing some habitat as well. But they are for sure the dominant. Yeah, Kate, you have to lead some Casey.
I forgot to put my birthday hat back on.
And you're going to have to share that with Casey.
I am absolutely happy to do that. I know she didn't pick it up when she came in.
I know.
To relieve me.
I know.
There's a thing out here on the ship, and that is if you are celebrating your birthday, you get to wear the birthday hat, which I have on my head at the moment.
It's quite attractive.
It is a
cake. It's a birthday cake with candles.
Well, I've got.
Looks like a top hat.
We're hoping that later on there's going to be an edible one.
Really?
Yeah.
Yeah, I don't. These things look like candles, but they also kind of look like some of the critters we've seen on the seafloor.
It's a sign of Cairo.
I don't understand why there's so much downforce. Maybe
it's just coming
across the
yeah.
ridge. And
So again, we're looking out. We can see.
And then just some with the hydrodynamics
corals,
of the vehicle.
sponges.
Yeah.
Well, doesn't it?
And the seafloor is this similar
move forward,
texture that we saw at the very beginning of the dive.
Like these ridges or lines of features.
That's
the same thing.
Yeah.
Yeah, so there's flat patches with probably fractures between the flat patches that have little ridges that look like they're right here in the video. It looks like they're maybe 10 centimeters, 15 to 20 centimeters high, of variable heights though along the ridges themselves. So micro topography. And the sponges really like the ridges.
And are oriented in the direction of the current as well, so that they can capture some particles.
The sponges,
yeah.
What's that thing? Center screen tile distance, kind of reddish pink. Have you seen that before?
Now it's a little pink coral on the left-hand side.
Yeah.
I think it's more bamboo.
Yeah, it looks like it. Boy, I'm getting good at this.
You are getting really good at this. We'll be able to change places soon. Oh, no, wait, I have to learn the rocks first.
Oh, man.
Looks like ship's about halfway through that 20-meter move.
Okay.
Look how straight the current is streaming the tether right now.
So what's that? Is that
That's another one of those red corals we saw earlier. It looks like it's got a brittle star
not nearly as much as I would have thought.
for dear life
I think
around the center of it.
Okay, Tara, let's
Yeah, it's fascinating to me that the brittle stars that like to live on these critters, they really tightly curl around them. So how do they get their food?
You got it.
So that would be a 1, 2. That would be a 3-fer.
That is amazing.
That's
that is a bubblegum coral.
Paragorgiid. A paragorgiid.
Popularly called brittlegum coral. If I'm right, Chris, on shore
coral.
in Hawaii. Is that true?
Yeah, makes you want to just chomp right down on that, and it has linguine next to it as well.
Hey, there we go. This is an Italian speaking
green linguine.
Well, I'm sorry, that other stuff looks like green papaya salad.
Okay, if you guys want to go up and down on that, that's great. Oh, that's me hanging on.
Wow, look at the little arm of that crinoid just wagging in the breeze.
All right, I'm steady now. On this.
All right, I'm starting to move toward you now.
Okay, can you tilt the camera down, Dan?
You got it.
So the stalk on this, if you go down
right
to
there.
the base of that, the stalk on that, one of the ROV crew just mentioned to me that the stalk on it looks like a lot like that tube that we saw at the beginning of the dive that was very fibrous. So it may be that what we saw at the beginning of the dive when we saw that very fibrous tube is the base of this Paragorgiid. This is great
I think that's
imagery.
a real possibility.
Yeah.
That's a good idea.
Full squat lobster on the edge.
This is a little oasis for all kinds of life.
Want me to tilt up, Joe?
Lovely
Yes,
little
please.
crab up there.
Yeah, you guys work the camera unless I
Yeah, that looks great.
have
Is that a
panic
squat
on this.
lobster or a crab? That's a squat lobster, right?
Yeah.
How quickly can you move
Oh, so
slope in this current?
Patty,
Yes.
Bruce said that a photo of the birthday hat is already on the Facebook. The underwater video site.
Lasers on.
Maybe find the lasers? Oh, there they are.
Just an oasis. We
Okay,
don't,
great.
Chris, we don't need a sample of that, do we? Or do we?
We did. I think we did get a sample of it
In leg
on
one?
leg
Leg one,
But
okay.
I'm not positive. And if Chris wants to chime in, that would probably be the definitive answer.
I would prefer to hold out for something glorious. It's just around the corner.
Okay,
There.
understood,
Yes.
understood. Because you know what'll happen. Then we'd have to have the pilot go in and pull one of those things out of the boxes. Just kidding.
Right, roger that. So we're looking for Coralis glorious.
Yeah.
All right. Then lifting up from here, turning left, and moving on.
So that crustacean that was on the bubblegum coral is Eumunida, which is the genus name, according to Mary Wixton.
Okay, let's go ahead and push ahead.
Pushing ahead. Thank you, Dan.
Seeing some sea stars and urchins. Another green eye. Lots of sponges.
Right. Wow.
Wow, pilot, there was a big one.
Holy smoke.
Actually, that looks different. I think that might be a geodeid.
Yep.
You want to
Yeah,
snap to
pilot.
that?
Yeah.
Yeah, can we snap into that big white, the
Yeah.
big white sponge, and
The
it's got
blob.
the blob, and it also has a.
That's
I
actually
think
has-
still have to keep flying. Get what you can
Hold
on.
that for
Interesting.
Huh. You know what that looks like to me?
What's it look like to you?
To me, that looks
Look
like
a
that.
helmet.
Kind of, but I was also thinking, remember that octopus that's the white octopus
Oh,
that
yeah.
looks that people call Dumbo?
Oh yeah, yeah.
Look at the Brittle Star City there.
Chris, what are you thinking? A geodeid, maybe? There's a kind of sponge that has, again, a very thick skin called geodia, like geodes. Okay.
Yeah.
Because when you crack them open, they have kind of
There are
layers.
crystals
Very
inside?
thick. No, I wish. They're silica spicules, though.
Okay,
Yeah,
silica's
yeah.
a good crystal.
Yeah.
Yeah,
Or
so
can
surely
form.
do the geodeids have to have those tufts coming off of them or not?
I'm sorry, say that again, Chris.
Do the geodeids, do they have to have those little tufts of spicules coming off of them or not?
Not. They do not. So this is a really interesting view because we're seeing at least 3 different species of sponges. The white filamentous mystery,
Great, that's good.
I was going to say animal, but it could potentially be plant. And the squat lobsters.
Let's go.
Lots of brittle stars. Lots of little brittle stars.
So could this be the Casper sponge?
Oh, I don't know. I don't know of that common name. Is Casper a very hard sponge?
The white octopus was dubbed Casper. I was thinking if it looks like the white octopus, we could call it the Casper sponge.
It could. We could call it the Casper sponge.
Is it a friendly sponge?
It is. They're all friendly. Some are, well, no, they're all friendly. Some are kind of smelly, so that would discourage friendship.
Are they friendly to the things they eat?
Well, see, the things they eat have even less of an ability to comprehend what's around them than the sponges do.
They get swallowed and go, where am I? Oh, this is nice.
So in a few minutes, we'll try and kind of turn on the mute buttons when this happens, but there's going to be a drill pretty soon.
There's
what are we looking
fish. Is that a fish? Is that a pizza fish?
No, there's another octopus. Oh, no. No, it's not. Oh, no, it's a fish. Oh, it's that sea robin. Yeah, cool.
Oh, right.
Yeah.
Oh, I saw pictures of this. Now I get to see it live and moving. Whoa. I love these guys. They're so cute.
So, yes, there's going to be a drill pretty soon, and so you may hear bells moving in the ringing in the background. So we'll try and buffer you from that.
It's just a drill.
And we don't have to do it.
It is not a genuine emergency. We're safe.
Yeah. Patty, you and I don't. We're excused, but later we're going to have to go and try on our Gumby suits. Oh, beautiful.
Tilt down. Holding.
It's called an armored sea robin, Scalicus or Satyrichthes. Satyrichthes or Scalicus.
So, Bruce, what can you tell us about this fish?
Yeah, why is it armored?
Oh, boy, you're putting me on the spot here, Chris.
Oh, you can just wing it.
Yeah, you're putting me on the spot here, Chris. This looks different than the ones that we have here in Hawaii because of all of these yellow spots on the body and stripes. Chris, have you seen any with this color pattern here in Hawaii?
No, this looks different to me.
Wow, here we go.
These are related
to move toward 350.
Yeah. These are related to the sea robins that are found in shallow water along the east coast of North America and down off Mexico.
Oh, that's beautiful.
They were placed in the same family as the sea robins for a long time, but now they're placed in a separate family because this group has these bony plates all over the body with the big spines sticking out that the regular sea robins don't have. You can see their thickened fin rays along the outer part of I think it's the pectoral fins sticking out in front of those fins. These fish actually use those thickened fin rays to walk along the bottom. They walk around slowly. And then the big fan-like fins, I'm not sure what they're used for, actually. They're not used for flying, like flying fish. This group also has these unusual prolonged bones on the snout that stick out like two horns. And again, the function of those is totally unknown. They've got large whiskers on the lower part of the head in front of the mouth that have a sensory function. So they walk along the bottom with those thickened fin rays, feel around the bottom for food items, and then that's how they capture small animals, small crustaceans, and other things. Not much known about the biology, though. And I think these are among the fish that have swim bladders that can move to produce sound, actually, make a drumming sound or a croaking sound. That's a little bit about what I know about them.
Bruce, do you think that the sound production is like an echolocation function for these animals?
Pilot, this is watched. You can keep moving, please.
To determine where they are on the seafloor?
Bridge, ROV nav.
ROV nav, this bridge.
We are ready for another ship
move. I don't know there's any evidence for that. My guess is probably not. It's probably more for another form of communication. Finding
good copy.
A potential mate.
In this low light environment. So another, probably more for communication.
No, I'm just getting going.
Okay. Well, thank you very much, Bruce. That's great to know. Wow. See, this is what's so wonderful about having the experts able to communicate with us on the ship, tell us what we're looking at. We would never know that without you guys. That's wonderful.
Are we going to go downhill according to HiPAC?
HiPAC has us going
Okay, we
uphill.
are currently at a depth
sure the
of
resolution of this
501
mapping data,
meters.
but I mean,